research, and my friend Elijah helped me with this research. The title of my slide, Variability and Antimicrobial Efficacy of Culturally Significant Indian Plants. So before I fully get started, I want to ask you guys a question. How many here have asked a question during a lecture or any kind of meeting, like just been confused and asked a question? Almost all of you guys. So I want you to hold on to that thought, where the idea is that questioning is actually an essential part of learning. It's the basis of learning. It's where you're engaging with the information, trying to understand it, kind of envisioning and opening up the darkness. So the idea is, I remember going to my first ever Bible study, and sitting there was a verse that was said that really stuck to me. It was in John 1:18 that said, "The light shines in the darkness, but the darkness has not understood it." This verse specifically stands out to me because it shows that in everything there is light. Light exposes what is hidden in the darkness, that is hidden in the depths of darkness, but it's what goes forward and kind of opens it up. I'd like to connect that to research itself. Research is essentially asking those questions, trying to discover that light, trying to open and understand what's hidden in that darkness. And that's what research is to me, and that's why I really like to engage with this research. This is my first ever independent research, but I really like to engage with this information as opening up to the darkness that is out there, that I've lived around all of these plants my whole life. I've grown up with these plants, but there's something that I wanted to discover more. I wanted to understand more about these plants. And the specific plants that I will be talking about is the curry plant, the drumstick tree plant, the sweet mint, and banana leaves. Specifically, these plants are so significant to this study because I've pretty much grown up with these plants. They're involved in so many dishes that I eat almost every single day. Not while I'm in college, unfortunately, but when I'm at home, like I'm with them, and I'm like dal, sambar, chutney, which is like a dipping sauce. It's just, I'm surrounded with these, but I never really know what is in it. I never really questioned it, because to me, my parents just told me this is significant, like these plants have exceptional nutritional values. Even research out there claims that these are exceptionally nutritional and have incredible medicinal values. So they're disguised as these perfect plants, but really, my question is, how perfect are they? There's research out there that has significant titles that really stood out to me. A review that says, a review on Mariah Kaningi, which is the curry plant, the miracle plant. Or the second one, where it says, Moringa oleifera, a natural gift. I think that's so significant because like they're saying that these plants are just so exceptional, are so incredible. And it's just, it really does stand out to me. Notice though, both of these researchers that say that these are these miracle plants, that say that they're a natural gift, are actually from India. It almost introduces some kind of bias. Like, could they be biased? Could they really be picturing these plants as something so incredible, even though they're just from India? So I want to kind of talk about all of this research, but this research on these plants is currently ongoing, and it's still active and ongoing. Much of the research much of the research in this talk on these plants is ongoing and active. And it's currently published in papers like International Journal of Trend and Scientific Research, many popular journals out there. So it's currently going, but there's a gap missing there. The gap is that they aren't comparatively together. So there's maybe 3 plants that are included, maybe 2, maybe 1 plant, but not all 4 of them together. So I wanted to do a comparative analysis between all 4 of these plants under the same environmental conditions. Along with that, another factor that I wanted to introduce, that was introduced to me by Dr. Ramesh, which was the variability in the bacteria. So there's 2 types of bacteria that I figured out, was the gram-negative and the gram-positive. The gram-negative bacteria is double membraned, so it's harder to penetrate for many active compounds, whereas the gram-positive bacteria is just single membraned. It's still significant, it's still a bacteria, but it's just there's a variability in the types of bacteria. The research out there is only testing either gram-positive or gram-negative bacteria, not both together. And that's the variable I'm including. That's the factor that I want to include in this study, where I'm testing both kinds of bacteria to really see how significant are they. So this is kind of how I laid it out. This is how my agar plates were laid out. So the odd numbers here will be your gram-negative bacteria, and the even numbers will be your gram-positive bacteria. I tested two types of the bacteria. So this one includes the gram-negative, so which was the E. coli and Serratia marcescens. And then we have the gram-positive bacteria, which will be your Staph epidermis, and then your Bacillus subtilis. And so within them, the positive control was the imbecillin, and then we tested all four of those plants to see how it flowed. Kind of want to dive into, then here are my results. So what I got was, I used the extraction process to really, oh my gosh, I think they skipped past the slide. Sorry about that. Yeah, okay, we skipped past this slide, but let me go back a little bit before I show you. You've already seen the results, but here's how I extracted those plants to really take out the active compounds that are present in the plants. So first, the leaves were grounded into a paste using the deionized water, and then I dissolved those compounds, or those extracted materials, into DMSO with 10% dimethyl sulfoxide. Now, DMSO is significant because it enhances the solubility between two types of compounds, or it enhances the phytochemicals that can come out of the extraction. So it allows wide ranges of chemicals that can come out of it and actually fight the bacteria. It makes it more efficient, and it allows the data to be more significant. Along with that, there was also, so after that, we just we created the extracts and we put them in the sterile disc and we tested the bacteria based on the agar plates that were mentioned here. So here are my results. So you can see that the banana was one of the only ones that kind of had that zone of inhibition here. And here you can see that there's a zone of inhibition that would be your zone of inhibition. It was approximately 10 millimeters. And that shows that comparatively, you can see the curry, the sweet mint, and moringa, none of them really had that zone of inhibition. Yet, they're framed in Indian journals and many journals out there as the most significant. Now, there could be many variables that could be the reasoning behind this, but it was definitely significant because it shows that the banana was the only one that was as significant. So, in conclusion, what I figured is the growth of these plants, the way they were grown, is different. So the exposure to chemicals that happens in India, so there's so many more toxins in the environment of India because of the Asian brown cloud. And so the plants there are exposed to various toxic chemicals, and they have to develop more nutrients to kind of fight that. So that could be one of the reasonings why the data of the plants there, it differs from the data I got. I definitely do want to redo this and try and find better results to see why exactly the curry plant, the moringa plant, the sweet mint didn't act as I expected, didn't act as the way I found the other results. Along with that, when I was doing the extraction and crushing up the leaves, I used deionized water. And that specifically would not have attracted or took out those active compounds that are found in a lot of the studies. So that could be one of the reasons what contributed in that study. Along with that, there was also methodological variability between my study and the way I did my extractions comparatively to other studies where they used dry leaves or they used essential oils. So that variability I do want to take an account into. Finally, I want to end on this note. Albert Einstein made an amazing quote that I really like to take in hand with, was that if we knew what we were doing, we would not call it research. And the idea is that we're just trying to figure it out. And the point of research is to ask those questions, to open that darkness, to figure out what's out there, to figure out what compounds are there. So that's the idea. And here are my references. Thank you so much. And if you have any questions, please let me know. Any questions? Okay. Yeah.
So you were saying that you have a lot of plants in your backyard, and I was wondering, like, if you were to use those plants
I used the plants that were in my backyard actually.
Okay,
So
well,
I used the exact ones that were grown in my
Okay,
backyard.
well, that adds on to my question. So, comparing like those plants, like plants in India, do you think like it's like how you were saying earlier, it wouldn't like give the same result? Like you need to have like plants from India to have, because they have like more resilience or resistance to like bacteria and stuff.
I would say yes, because you have to take into account that the exposure in different environments can introduce another variability. So different environments can elicit different kinds of compounds within plants, and that could be a part of why what compounds come out of the plants are different. That could also mean why some of the plants that are in India might be more effective, more antimicrobial than other plants. Does that answer your question?
Yeah.
Okay. Yeah. What's your question?
What exactly were your expected results at the beginning, and how did that differ from the results that you got, or like how did that differ from other research studies that were conducted?
Yeah. So my expected research, because my parents I don't know if you relate to this. My parents have idealized these plants, and like to me, I was like, these plants have to find at least have some kind of zone of inhibition. So I didn't really do any research on what exactly the zone of inhibitions would look like, but I expected at least something. But as you saw, all it did was stain the area. It didn't really fight any kind of compounds. It didn't really do anything. So I think that really surprised me. Does that answer your question? Awesome. Yeah. Go ahead.
I just wanted to ask, do you think doing more research studies like this can help us kind of solve the issue of nutrient loss nutrient loss that we have here in America? Because I know that some farmers, they use a lot of pesticides, or they use ingredients that reduce the content of nutrients in some of our fruits and vegetables. Do you think having more studies like this, comparing American grown food and let's say fruits and vegetables grown in Mexico, can kind of help us kind of go around?
I would say yes. That's a really good point. I think that there is variability between environment and what they're exposed to. So there could be, but I feel like if it's in the same continent, it might not be as significant. Introducing those pesticides and those different chemicals, that will definitely elicit some kind of, especially GMOs, like they will have different kind of compounds or different kind of effects. However, like comparing it to somewhere where, like in Mexico versus the United States, those kind of comparisons might not differ as much as maybe United States with, let's say, India or Africa or, the different continents. Does that kind of answer your question? Yeah. So yeah, I would say that there will be variability between the continents more than it will be just regions or countries. Yeah. Any other questions? Okay, thank you so much. Thank you for being